Let me take this opportunity to invite Noutin to come and start his presentation. Let's all give him a warm welcome while enjoying those treats. Thank you.
As you can see there, my name is Noutin, basically, and my talk will try to embrace contribution of synthetic biology in phage therapy. So, as we all know, there is issue of resistance. Bacteria strains all over the world. It's not only bacterial resistance all over the world. There is also parasite resistance issue. Here we are going to point out antibiotic resistance problem. So, and we believe that if there is no really innovation, and it has been reported and projected that about 10 million deaths will occur by 2050 in all over the world, but in developing countries, including Africa, about 90% that will occur, and we're gonna have a huge economic impact. So there's need for innovation, and one of the innovative way that we are going to usually use the word resurrect or revive is phage therapy. So what's basically phage therapy? I want to make an analogy very fast that we know that SARS-CoV-19 does damage our lung cells. So, and hepatitis B virus does damage liver, isn't it? So, it means each virus has its hosts. So, when it's come to bacteriophages, their hosts are basically bacteria. So, what they do is that they will try to find their specific host bacterium, then absorb to that, inject their nucleic acid DNA, and then hijack the um, replication machinery of the host bacterium in order to lead to its complete destruction. Yes, many studies have been done, and for phages to be used as product, it has to be double strand DNA, means belong to the order of Caudoviriales and bacteriolytic. There are other therapeutic parameter when it's come to use of phages as therapeutic agents. Yes. So when we look at all those studies, you see that there's a kind of gaps, and the major one is there's no identification of phages with therapeutic markers against resistant bacterial strains, so we have bacteria of clinical importance, especially in Africa. So, but also there is lack of innovation when it comes to synthetic biology that will try to bring it out. Yes. So as I said, we need to know the host bacterium of the phages. So far we have been conducting a work on isolation of Klebsiella or carbapenem resistance Klebsiella pneumoniae, and you see on this figure that we have been able to identify one of the highly resistant genes called new jelly metallo-betalanase genes. So it's carbapenem genes. So we have published something on that, and you can look it up. Yeah. So let's. Yes. let's talk about phages. So to obtain phage is very simple. Yeah, you can see me dressed very nice here, but when it's come to work, we do change the attire. So you go to the field whereby you can get it from sewage samples, and as you get it from sewage samples, you're gonna use a number of techniques which will enable you to isolate the phages. And here is the phages act like basically um, a lytic agent. They act like antibiotics, whereby you are going to observe a clearing zone if they are really specific to the host bacterium of your interest. So that's what happened here on the second image. You could see there is a clearing zone. We call it normally spot, and in that spot there is a bunch of phages, and you need to purify those phages at least get to try to get a single one through placacy. That's what we have done, and next slide, you see there is a purification of those pure phages, whereby we use sodium chloride and polyethylene glycol in order to come up with a pure phage lysate. And you can see on the third figure that there are append of tubes there. In those append of tubes, we are having a clear lysate. We get those precipitated, they contain precipitated phages. We get it from normally the first figure that you are seeing on the figure 3. You can see that there is a kind of lytic activity going on there. There is one which is really a turbid, and there is one which is clear there. So see how we have managed to get this small quantity of phages or precipitated phages, and we try to uh, find out whether they are still viable, and it was the case. They are still viable, and their concentration is about 10 power 9 platforming units per ml. Yes, so as you get your phages, you need to know what it is. So we went for morphological characterization of it using transmission electron microscope, which is not really available in Africa. That's one of our challenges. But we managed to perform this analysis, and you could see that it really belongs to the order of Caudovirales. So we have a therapeutic property. So we went for whole genome sequencing of the phages and using nanopore sequencer, and you could see that nice figure there. We are trying to finalize bioinformatic analysis. Yes, so it's basically when you have phage genome, it has hypothetical proteins, also tail proteins, and capsid proteins. People make use of their hypothetical proteins in order to come up with synthetic or synthetic phages. So as we have gotten that phages, we need to find out, we've been trying to find out their efficacy, and one of the tests that we use is the host range analysis. So we try to find out how specific those phages are. So you can see we have a number of phages, and from M0, there we have cocktail, call it cocktail phages. It's a combination of more than two phages in order to have a better performance. So when you look at those figures, we have different strain, Staphylococcus aureus strain. We have also E. coli strains, and you will see that our phages are really specific to carbapenems produced in Klebsiella pneumoniae. And you will see also there is a lactobacillus there, which is one of the microorganisms which belong to the normal flora. So we see that there is no really our phages are not sensitive to those to that microorganism. So that's what I want to highlight there. So, Yeah, so still in line with the efficacy of phages that you have isolated, you need to compare it with existing antibiotics, both the one which is not sensitive and the one which is more effective. And here we have imipenem and tigecycline, and you see the performance of phages when it is combined with imipenem and also with tigecycline. And the one in green, you see that there is a synergistic uh, actions between imipenem and our cocktail of phages. Yes, it means we know very well the mode of action of phages. So after destruction of the bacterial cells, imipenem will have its sites of binding in order to also act as an antibiotic. So that's what people are really looking for. Uses phages as adjunct therapy. Yes. I would like here to before talk explain that slide. I would like to tell you that I feel very happy to be before you this morning and present something of phages because my first project was on isolation and molecular characterization of phages against Mycobacterium tuberculosis, and it has been really challenging for me to move forward that project. It has not been approved, and as I've been saying, you need to have really a strong team before really using synthetic biology. Yet people are working on the use of chimeric phages or synthetic phages um, to improve the performance of phage therapy. And I would like to bring out the work of Diana Pires. She's a postdoc in Portugal, doing incredible work, and she has managed to improve the therapeutic parameter of the phages by inserting what we call the GP12 protein or genes into the wild type phages, and the GP5 protein or 12 protein are basically the tail proteins of T4 phages, and when she has done it, she has used a technique called bread. And for more detail, you can look up that paper to know more about the bread. Those who may have more insight in synthetic, the different methods used in synthetic biology. So yes, if you are not able to use phages in therapy, you can also use it in diagnosis, and people are using phages in order to improve cancer therapy as well. And we can also use it as a disinfectant. There are many other applications. Yes. So I would like to acknowledge all those people who have been supporting me, from Pan African University to JQuat to Institute of Primate Research, but also the Phage from Phage Directory, and the team was giving me the opportunity to present this. Merci beaucoup. Many thanks. Santé. Mwebali.
Thank you so much, Newton, for that great presentation about phages. Yes, I can allow one question from Harry.
I was going to ask you, you mentioned that you had a challenge implementing the phage, synthetic phage project. What was specifically your challenge to implementing that?
Yes. As I said, normally I'm from Pan African University, and the thesis board, before approving your project, they need to know whether you have someone who have already worked on phages, especially in the coming of the synthetic phages, But also the host bacterium I was about to use was Mycobacterium tuberculosis, which you should know the biosafety level required for that. Yes, I have safety level 3. It's also go for 4. So I couldn't find it, and that really was about to work at January, but COVID-19 came, so but take it off. So that's where I went to now. It's the primate check because you can work from anywhere as a Pan-African scholar. And over there they don't use it so much as well. So I need to find out how to move forward. But I believe that I can make it one. There's no issue for that.
So, if I understand you correctly, it's not more of it's not more of a technical challenge to that is preventing you from implementing this work. It's more of access to a platform to enable this work. Okay.